What is the exact role of oxaloacetate in the citric acid cycle? Oxaloacetate is a 4-carbon molecule formed from aspartate via transamination reaction or from the malate of TCA cycle. Oxaloacetate presence is very much required to initiate the citric acid cycle. This is what reacting with the acetyl CoA and making the citric acid compound, further which undergoes oxidation and decarboxylation and releases NADH and FADH2 from the TCA cycle. The oxaloacetate acts as catalyst. It participates in the oxidation of acetyl group. It is helping to extract the energy from the carbons and hydrogens of the acetyl group, but it is self-regenerated at the end of the TCA cycle. Thus, one molecule of oxaloacetate is capable of participating in many TCA cycles. One molecule of oxaloacetate is capable of participating in many TCA cycles in many oxidation of acetyl molecules, and oxaloacetate, that is the reason why it is regenerated back. It is simply acting as a catalyst, but its presence is very much important to initiate the TCA cycle. Without oxaloacetate, you cannot extract the energy from the acetyl group. You cannot incorporate the energy from the acetyl group into the NAD plus and FAD molecules. So no generation of reducing equivalents, which will disturb the cellular respiration. Let's start discussing the citric acid cycle step by step. As I have emphasized the formation of citric acid many times from the union of oxaloacetate and acetyl CoA. Oxaloacetate, again, it is formed from the aspartate via transamination or from the malate in the TCA cycle. Acetyl CoA has many sources, as we discussed. It can be produced from fatty acids, ketone bodies, amino acids in the starvation period. Normally, it is generated from pyruvate molecule within the mitochondrial matrix via pyruvate dehydrogenase catalyst. These two molecules, oxaloacetate 4-carbon compound and acetyl CoA 2-carbon compound, condenses together to form citric acid. So the type of chemical reaction during condensation is aldol condensation. That is, oxaloacetate contains 2 carboxyl groups and a enol group here within its structure, and acetyl CoA contains a CoA group and a C double bond O enol group. While condensation, the C double bond O of oxaloacetate reacts with the methyl group of acetyl CoA and leads to the formation of CHO aldehyde group. That's why it is called aldol condensation. The union of methyl group of acetyl CoA with the enol group of oxaloacetate is forming into aldol group in the citryl CoA. Citryl CoA, with the help of citrate synthase, it is converted to citric acid having 3 carboxyl group in its structure and 6 carbon molecules in its structure. 1, 2, 3, 4, 5, 6. 6 carbons. Along with 6 carbons, there are 3 carboxylic group. 1, 2, 3. Let's move forward into step 2 of citric acid cycle, that is formation of isocitrate. Citrate is isomerized into isocitrate to enable the 6 carbon unit to undergo oxidative decarboxylation. In chemistry, isomerization is a process which helps the functional groups or reacting groups of the molecule to go near to the reactant site so that the chemical reaction takes place easily. The isomerization of citrate, same way, helps to undergo the citrate molecule a couple of oxidation, reduction and decarboxylation reactions taking place in the TCA cycle. In the second step starts with dehydration, removal of water molecule followed by a hydration step. The result is an interchange of hydrogen atom and a hydroxyl group. In the structure of citrate, we see the interchange of hydrogen atom and hydroxyl group. Hydrogen and hydroxyl group, they interchange their position between second and third carbons of citric acid molecule to favor the further chemical reactions in citric acid cycle. The enzyme catalyzing these steps is called aconitase. It is named aconitase because there is a compound, intermediate compound formed between the citrate and isocitrate complete transformation, that is aconitase. So here we can see the hydrogen atom linked to the second carbon and hydroxyl group linked to the third carbon are interchanged. That's why citrate is converted to isocitrate. Citrate and isocitrate, these two are isomers. Isomers, in these two isomer forms, the hydrogen atom and hydroxyl groups links to second and third carbons have interchanged their position. In between the transformation into isomer isocitrate, there is also formation of cis aconitate, based on which the enzyme catalyzing the reaction is called as aconitase, which is an iron sulfur protein, which is a non-heme protein because it contains iron but it doesn't contain porphyrin groups linked to the iron atoms in the structure. Let's elaborate step 3 of citric acid cycle, that is formation of alpha ketoglutarate from the isocitrate. Isocitrate, in order to transform to alpha ketoglutarate, it undergoes dehydrogenation and decarboxylation with the help of isocitrate dehydrogenase enzyme. The decarboxylation process requires magnesium or manganese ion as cofactors. In total in the cell, in an eukaryotic cell, there are 3 isozymes of isocitrate dehydrogenase. Isocitrate dehydrogenase linked to the mitochondria generally uses NAD+ as the cofactor. The other two uses NAD+ as the cofactor, and the other two isozymes using NADP+ as cofactors are found in the mitochondria and in the cytoplasm. The one which is linked to the TCA cycle mainly utilizes NAD+. So hence we see NADH generation in the TCA cycle. We only have NADH generation in the TCA cycle because the enzyme participating in the TCA cycle utilizes NAD+ as the cofactor. Especially the isocitrate dehydrogenase molecule is linked to the NAD+, which involves in TCA cycle. Therefore, we only see NADH formation in the TCA cycle. The other two isozyme forms which utilizes NADP+, they are not taking part in TCA cycle at any time in the cellular respiration. So here we can see the isocitrate, which is transformed to oxalosuccinate and finally to alpha ketoglutarate. Here we have respiratory chain linked oxidation of isocitrate proceeds almost completely through the NAD+ dependent enzyme. Here we have dehydrogenation. The enzyme having suffix dehydrogenase, there we have most of the time generation of NADH or FADH2, mostly NADH. So the enzymes linked to NAD plus cofactor, they always generate NADH. Wherever there is dehydrogenation, there is extraction of hydrogen atoms from the substrate into the NAD plus, resulting NADH plus H plus formation. Isocitrate is modified to oxalosuccinate intermediately, and oxalosuccinate undergoes decarboxylation into alpha ketoglutarate. The carbon compound from the oxalosuccinate is removed and 5 carbon molecule alpha ketoglutarate is synthesized within the TCA cycle. Let's elaborate on the step 4 of TCA cycle, that is formation of Succinyl Co A. The important thing to be noticed here are the alpha ketoglutarate transformation to Succinyl Co A is unidirectional. Once the Succinyl Co A is formed from the alpha ketoglutarate, never again Succinyl Co A is turning into the alpha ketoglutarate shape. Same with isocitrate conversion to alpha ketoglutarate. Once the isocitrate is transformed to the alpha ketoglutarate, it's not again turning back into the shape of isocitrate. So these two steps, the step 3 and step 4 of TCA cycle, can be considered as rate limiting steps which maintains the flow of TCA cycle chemical reactions. The conversion of isocitrate into alpha ketoglutarate is followed by a second oxidative decarboxylation reaction, that is formation of Succinyl Co A from alpha ketoglutarate. Here also we have release of carbon dioxide and generation of NADH, same like in the previous step of the TCA cycle. So the alpha ketoglutarate, which is 5 carbon compound, by decarboxylation it is transformed to 4 carbon compound, and there is generation of NADH also as like the step 3. Alpha ketoglutarate undergoes oxidative decarboxylation in a reaction catalyzed by multi-enzyme complex similar to the pyruvate dehydrogenase complex. Pyruvate dehydrogenase plays an important role in converting pyruvate into acetyl CoA molecule to start the TCA cycle. Alpha ketoglutarate dehydrogenase, which is playing important role in the intermediate steps of TCA cycle, the structure of alpha ketoglutarate resembles the pyruvate dehydrogenase as they do the same job, that is dehydrogenation. They do require, both of them require the cofactors thymine, NAD, FAD and coenzyme A. If you remember, I have already emphasized the importance of these cofactors. From what are the sources from where these cofactors are derived? These cofactors are derived from vitamin B complex. If the person has vitamin B complex deficiency, it will impair the TCA cycle by impairing the function of pyruvate dehydrogenase and alpha ketoglutarate dehydrogenase, as they are dependent on these molecules for their performance in the TCA cycle. We are at the step 5 of TCA cycle, that is formation of succinic acid. Succinic acid is forming from the substrate Succinyl Co A. Within the Succinyl Co A, there is an energy-rich bond, that is thioester. So, how to extract this energy present in the thioester bond of Succinyl Co A? We can do this by substrate level phosphorylation. What is substrate level phosphorylation? extracting the energy within the substrate by phosphorylation reaction. Carefully listen. We are extracting the energy present in the substrate by phosphorylation reaction. That's why it is called substrate level phosphorylation. And also it is called substrate level phosphorylation because generally we see the synthesis of ATP at the oxidative phosphorylation pump. But here we have the synthesis of GTP or ATP in the chemical reaction itself. That's why it is called substrate level phosphorylation. There the motivator for the generation of ATP is proton gradient and proton flow into the oxidative phosphorylation pump. Here directly the energy present within the substrate is extracted into the GDP or ADP molecule. That's why it is called substrate level phosphorylation. And who is catalyzing this step? The enzyme which is catalyzing is Succinyl Co A synthetase or Succinate thiokinase. Here we can see Succinyl Co A. The Co A bond is removed. The Co A is removed from the Succinyl Co A and the energy stored here in this molecule is released into GDP. GDP is converted to GTP. This is the only example in the citric acid cycle of substrate level phosphorylation. Tissues in which gluconeogenesis occurs, they contain two isozymes of succinate thiokinase. One isosome form is linked to GDP and another isozyme form of succinate thiokinase utilizes ADP as the cofactor. That means one succinate thiokinase makes ATP and another isozyme form of succinate thiokinase mainly associated with TCA cycle is making GTP. Mainly we are extracting the energy present in this thioester bond of succinyl Co A molecule in this bond, succinyl Co A bond into the GDP and making GTP at this chemical reaction. We are extracting energy from this substrate and making GTP. That's why it is called substrate level phosphorylation. So, how do we use GTP? Where do we use the GTP formed in the TCA cycle? It is used for the decarboxylation of oxaloacetate. So, TCA cycle have two roles in the metabolism. TCA cycle is mainly breaking down the acetyl CoA, generating an ADH, finally linked with the energy generation at the end of electron transport chain. And also it has anabolic role. The intermediate compounds are precursors for many other compounds, molecules in the cell such as amino acids and carbohydrate molecules like pyruvate, glucose, etc. So in the times of starvation, the oxaloacetate formed at the end of the TCA cycle can be used for gluconeogenesis. So the oxaloacetate is converted to phosphoenol pyruvate. So for this reaction, the energy is required in the form of GTP. Hence, the GTP formed within the step 5 of TCA cycle is participating in gluconeogenesis process and provides a regulatory link between citric acid cycle and the withdrawal of oxaloacetate for gluconeogenesis. So as gluconeogenesis tissues contain both isoenzymes using GDP and ADP as the cofactors. What about the non-gluconeogenic tissues? They have only one type of isoenzyme form of succinate thiokinase that uses ADP as the cofactor. So we can expect only the formation of ATP in the non-gluconeogenic tissues TCA cycle.